Where's Sami? Oh. Ah, shoot. All right, girls, are you ready?
1 second, I'm just gonna.
Okay, just unmute yourselves. And
Can you see my screen?
Yeah.
Okay.
See the slides, all good.
okay.
Okay. I think I'm ready.
Alrighty. I am just working on my windows here. Okay, we still have a couple people coming in. All right, let's get started. Welcome to Eliminating Imperfections or Ethical Interventions, the Ethics of Gene Editing and Prenatal Screening, presented by Sami Sharp and Elizabeth Weichner. My name is Kim Pearson, and I will be the moderator for this session. Please note that this Zoom session is being recorded. And if you haven't already, please mute yourself. In addition, we recommend that your screen is in speaker view. That's located in the top right. During the presentation today, you have access to the chat feature, which is indicated at the bottom. And if you would, please share any questions during the session privately with me. And I will share them when appropriate. And please note there will be time at the end for questions. And with that, I'm going to turn us over to our presenters.
Hello, I'm Elizabeth Weichner and my partner is Sami Sharp. Our presentation for the Bioethics Symposium is Eliminating Imperfections or Ethical Intervention, the Ethics of Prenatal Screening and Gene Editing. For my part of the project, I looked at gene editing for HIV and AIDS with a focus on religion. I looked at 5 different religions, Christianity, Judaism, Islam, Buddhism, and Confucianism, and use what the religion said about gene editing to guide my ethical analysis.
Hi everyone, my name is Sami, and for my side of the project, I studied the modern usage of prenatal screening technology, specifically for Down syndrome, and I studied the consequences of prenatal screening through different stakeholders' perspectives, and ultimately by analyzing whether these technologies could lead to a new eugenics movement. So to begin, we're going to launch a few poll questions just to get everyone's initial feelings on these topics. So you should see a poll. So the first question is, would you use gene editing to create an HIV-resistant embryo?
What would you say, Cass? No. Would you screen your child for Down syndrome?
Our next question is, would you screen your child for Down syndrome?
Next, would you be okay with living in a world with genetically modified people?
And our last question is, would you be okay with living in a world without genetic diseases?
So
We'll give you a couple minutes to look this up. Okay, we're getting close, but we'll still give you a couple, like 30 more seconds. Okay. I think that we could end the poll and we can go over the results. We're going to share them so that everyone can see what everyone thought.
So you should be seeing the results. So keep these in mind as we go through the rest of the presentation and see if your initial reactions change as we share a little bit about our projects.
So with that, we are going to transition into our overall ethical question, which is, what are the ethical considerations in using gene editing and prenatal screening? We'd just like you to keep this question in mind as we go more into our ethical analysis, but right now we're going to transition into the science background part of our presentation.
So firstly, from my side, prenatal screening tests are a set of procedures that are performed during pregnancy on expectant mothers to determine whether a baby is likely to have certain birth defects. And there are several different forms of prenatal screening. Firstly, chorionic villus sampling is an invasive screening test that involves taking a small piece of tissue from the placenta. Next, amniocentesis involves sampling the amniotic fluid, which surrounds the baby during pregnancy, and it contains fetal cells with the genetic makeup of the baby, and it's removed from the uterus for testing. But lastly, the most accurate form of prenatal screening is the AFP blood test, and this can be performed between the 13th and 27th week of pregnancy, and it provides a successful detection rate of 80%. And one of the things that prenatal screening can detect is Down syndrome. There are 3 forms of Down syndrome. 95% of the time, though, Down syndrome is caused by trisomy 21. And this is when a person has 3 copies of chromosome 21 instead of the usual 2 copies in all cells. And this is caused by abnormal cell division during the development of the sperm cell or the egg cell. As a result of this, the developing fetus will receive an extra dose of the product of hundreds of genes, which alters the delicate balance of proteins and other chemicals in the body.
I looked at gene editing for HIV and AIDS, but before we dive into what gene editing is, we need to understand what HIV and AIDS are. HIV stands for the Human Immunodeficiency Virus, and it's a virus that works by infecting CD4 cells in the immune system. HIV works by bonding to the extracellular protein receptors on the outside of CD4 cells. It uses the RNA buds that you can see coming off of the purple virus in the middle of the picture to connect with the CD4 cells. Once it bonds to the CD4 cells, the virus can either kill the cell or trick the cell into producing more of the virus. Something that is particularly interesting regarding HIV is that it can only be transmitted from person to person through a contact of bodily fluids. This is most common during unprotected sexual intercourse or sharing needles either during drug use or in underprivileged communities. This is a large contributor to the stigma behind HIV and the discrimination that those who are HIV positive face. Because of the way that HIV is transmitted, it also brings up the question of should those who are HIV positive have a responsibility to tell others that they have HIV in order to protect the greater good. I'd just like you to think about this question as we continue our discussion. The long-term effect of HIV is AIDS. AIDS stands for Acquired Immune Deficiency Syndrome. Someone who is already HIV positive can be diagnosed with AIDS when their CD4 cell count drops below 200 cells per milliliter of blood. To put that into perspective, the ideal CD4 cell count is between 500 to 1,000 cells per milliliter of blood. We are currently living in the AIDS epidemic. There are more cases of HIV and AIDS than there have ever been before. Currently, there are 37.9 million people in the world who are living with HIV or AIDS. However, there are probably more who are unaware of their infection. This is because 14% of those who have been infected with HIV within the past year are unaware of their infection. This is because it takes some time for the HIV virus to hurt the immune system enough for the individual to feel the effects. And if an individual does not know that they have come in contact with HIV positive blood, they do not even know that they need to get tested. And the AIDS epidemic is still growing because 1.7 million people were newly diagnosed with HIV in 2018. So scientists are looking for a solution to this epidemic. They think they might have found it with gene editing in CRISPR-Cas9. Gene editing is a form of genetic engineering. With gene editing, scientists can manipulate the human genome by adding or deleting certain bases in order for an organism to have a desired trait. Although there are many different types of gene editing, I'll be focusing on CRISPR-Cas9. CRISPR stands for Clustered Regulatory Interspace Palindromic Repeats, and Cas9 stands for CRISPR-associated cascade protein 9, which is an enzyme that will cut the DNA. Gene editing for HIV resistance is used to target the CCR5 gene. All gene editing for potential HIV-resistant humans has to be done on embryos. These embryos need to be created through in vitro fertilization, which is where the embryo is created outside of the body. This is because CRISPR is a procedure that happens outside of the body. Gene editing for HIV resistance will target the CCR5 gene, which is located on chromosome 3, position 21. The CCR5 gene codes for the protein extracellular receptors on the CD4 cells. So scientists think that if those receptors are gone, HIV will not be able to infect the cells, even if it has entered the bloodstream. To do that, scientists need to create a strand of guide RNA that has the complementary base pair sequence to that of the CCR5 gene. That guide RNA will help the Cas9 enzyme find the correct place on the genome to cut the DNA. That guide RNA is incorporated into the Cas9 enzyme, and that complex is incorporated into the embryo. Inside the embryo, Cas9 will split and scan the DNA and search for a sequence that complements the guide RNA. When it finds that sequence, Cas9 will conform itself around that piece of the genome and pull it away from the rest of the genome. This will induce the delta 32 mutation. This is a mutation that is a naturally but rare occurring mutation that is known to result in HIV resistance.
So now we have some scientific background on our topics. We have 2 more polling questions just to get your initial reactions. So which technology, gene editing or prenatal screening, do you think could do more harm? And which technology, gene editing or prenatal screening, do you think could do more good? Okay, we'll keep going for about 10 seconds and then we'll end the poll. Okay, there you go. So I'm gonna share the results. So that's really interesting, these results. You can see that with the first poll, a lot more people thought that gene editing would do more harm, but in terms of doing more good, we're a lot closer to half and half. So that's super interesting to think about as we keep going through more of our ethical side of our presentation.
So with that, we're going to transition into our first bioethical principle that we're going to be talking about, non-maleficence. Non-maleficence means do no harm, and it is important to consider with prenatal screening and gene editing because the last thing we want to do is cause harm. In regards to non-maleficence and gene editing, the first issue is that the long-term effects are unknown. Gene editing technologies are such a new technology. It was only harnessed for human use in 2007. Few people are willing to use gene editing technologies until we know what could happen to a genetically modified human down the line. In my opinion, however, the more important idea in non-maleficence is inaccurate gene cutting. The human genome is over 1 billion base pairs long, and because it is so long, sequences repeat themselves throughout the genome. And when the guide RNA is fairly short, and in the case of editing out the CCR5 gene, the guide RNA is only 32 base pairs, that sequence is likely going to repeat itself multiple times throughout the genome. And whenever the guide RNA finds a sequence that matches it, it is going to pull it away from the rest of the genome. However, that sequence might not be the true CCR5 gene, it might be part of a larger gene. This could cause necessary genes to turn off, which could lead to the embryo dying. One of the religions that I looked at was Buddhism, and one of their main principles is to avoid killing at all costs. If you look at inaccurate gene cutting in Buddhism, it would seem that Buddhism would reject gene editing because they see it as a way of doing potential harm, because if there is inaccurate gene cutting, the embryo could die. Although this is only looking at one lens of Buddhism, it would lead to the conclusion that Buddhism would not approve of gene editing technology.
One of the key issues of non-maleficence in prenatal screening for Down syndrome is the issue of quality of life. Nowadays, treatments such as cardiac surgery, vaccinations, antibiotics, leukemia therapies, and anticonvulsive drugs have considerably improved the quality of life and life expectancy of individuals with Down syndrome. Therefore, it's important to analyze if you are doing harm by preventing the birth of a life with a disease that is becoming more and more treatable and less detrimental and harmful. And physicians and parents have to recognize what quality of life they believe is acceptable, and whether there is ever a quality of life that can be universally regarded as acceptable. Furthermore, physicians have to weigh the importance of the potential benefits or harms to one's quality of life when using prenatal screening for Down syndrome. Next, we're going to transition to another ethical principle of autonomy. And autonomy can be defined as the ability of a person to make his or her own decisions. When discussing autonomy with prenatal screening for Down syndrome, a key issue is whose decision is it to make choices for a potential child and who can decide if you're doing harm. So a big part of my project was understanding and studying the importance of parents as stakeholders and how to ensure their autonomy in these decisions. Prenatal screening is revolutionary because it gives prospective parents the ability to make informed, private decisions about parenting a child with Down syndrome. And with the availability of legal abortion, it allows them to exercise their autonomy over decisions about a potential child. However, on the other side, genetic counselors were a large stakeholder that I studied in my project. And they play a key role in prenatal screening because they inform parents on the diagnosis of Down syndrome. But in order to understand genetic counselors as a stakeholder, we have to consider how the social construct and view of those with Down syndrome could implement implicit bias into genetic counselors' advice. My research has shown that this implicit bias can be implemented into advice just through a negative tone being used when telling a patient that prenatal screening shows their embryo has Down syndrome. For example, they might use phrases such as, "I'm sorry," or "Unfortunately, I have bad news for you." Also, these conversations can void of the positive reality that many individuals with Down syndrome can become semi-independent and live with good medical care far into adulthood. So overall, it's important to understand how genetic counselors can influence the decisions of their patients and how they can promote the act of preventing the birth of those with Down syndrome through implicit bias and how that can implicitly restrict the autonomy of parents.
Part of the job of my bioethics project was to propose my idea for what regulations on gene editing technology should look like. However, all regulations, by definition, limit autonomy because people would be guided by what the law says they're allowed to do rather than what they personally would choose. One of the religions that I looked at was Christianity, a religion that has many times stated that they are against gene editing because they see it as a form of playing God. However, even if gene editing was allowed in the eyes of Christianity, they would like any decisions about whether or not a parents are allowed to use gene editing technologies to be made by the church and directed by God. In a way, this is taking away the individual's autonomy and their right to decide what to do with their child's embryo.
So after hearing two of our main ethical principles, our last poll question is, in your opinion, which principle, non-maleficence or autonomy, do you think is more important? Okay, we can share the results. It looks like it's coming to an end. So interestingly, you can see that more people think non-maleficence is important, but it's very close. So keep that in mind. We're going to transition to a value now, but that might be important when it comes to all case studies later. So now we're going to transition to a key value in our projects of equality. In my project, equality is tied into understanding the value of those with Down syndrome. Firstly, with prenatal screening for Down syndrome, we have to think about the messages that we're sending about the value of those with Down syndrome in our society. But also, the value of equality raises the question of to what extent are a person's genetic origins essential to their identity? And if a person's genetic disorder is considered a part of their identity, then isn't the value of equality at risk? Because this idea that we're all equal in human value, regardless of our background, is being challenged. Another aspect of equality in regards to prenatal screening is issues of accessibility to technology. And looking into the future, if these new technologies are only readily available to the wealthy, that could result in a lower class that will still suffer from inherited diseases and disabilities. Additionally, a big part of my project was analyzing different nations' rates of prenatal screening usage and their perspectives. And a big disconnect that exists is between nations with systemized healthcare and those without. For example, nations like the UK give a wider availability of these technologies to their citizens because they have the National Health Service. And therefore, this could and has been leading to certain nations having rates of children with Down syndrome rapidly decreasing.
$4,500. That is how much it would cost to genetically modify one gene of a child's embryo in the United States as of 2019. While this number is significantly lower than many would think, $4,500 is still a large enough number that public use of gene editing will create a divide between those who have access and those who do not. And this number does not tell the full story. Any embryo that needs to be eligible for CRISPR-Cas9 gene editing needs to be created through IVF, which can cost up to $12,000 for just one round. And most insurance companies do not cover these costs, so people will likely have to pay out of pocket. However, the price of gene editing has significantly decreased within the past year, and now scientists can use gene editing in a lab on E. coli cultures using a kit that costs just $99. And as more people start to use gene editing technologies, the cost will likely decrease, allowing more people to have access. Another important aspect of equality is the geographical divide between where the largest HIV positive communities are located and where gene editing is available. On the screen is a map showing the percentages of each country and their HIV positive communities. The largest percentages are found in Southern Africa, particularly in the countries of South Africa, Botswana, Lesotho, and Swaziland. There's also a large percentage in Russia. However, the locations where HIV is most prevalent does not align with the countries that have access to gene editing technology. This map shows where there are legal regulations on gene editing technology. And if a country has legal regulations, it means that they do have access to gene editing technologies. Gene editing is most developed and accessible in Brazil, the United Kingdom, Japan, Germany, and the US, with China being the world leader in gene editing technologies. This does not align with where HIV positive communities are located around the world. And those communities are likely the ones that would benefit the most from gene editing for HIV resistance. It is also important to note that many countries on this map show that they have a ban on gene editing or restrictive rules. Even though China has a ban and guidelines on gene editing, those rules have been violated many times. We'll delve into this idea more with the experiments of He Zhonghui in our case study later on. One of the religions that I looked at was Confucianism, and they would argue that because gene editing promotes inequality by having a divide between those who have access to it and those who do not, it would be seen as unethical in the eyes of Confucianism.
Next is our last analysis of the overall theme between our projects of the slippery slope. Both of these technologies of prenatal screening and gene editing can create a slippery slope in society. Firstly, we could get carried away correcting perfectly healthy babies. Once we start down the slippery slope of eliminating embryos because they're diseased, what is to stop us from picking babies for their physical or psychological traits? Furthermore, we have to analyze the intent we have in editing and preventing the birth of embryos with certain abnormalities. This idea of our intent was a large part of my side of the slippery slope theme, and it led me to question if prenatal screening was slowly creating a new eugenics movement. For some background, the eugenics movement was in the early 20th century, and it targeted those who were seen as imbeciles, the feeble-minded, criminals, and the poor, and these labels reflected racist and classist biases and judgments. So when analyzing our intent, we have to ask if Down syndrome is regarded as objectively bad and undesirable, and therefore is society encouraging prenatal screening to detect Down syndrome because we still adhere to the attitudes that prevailed in the 20th century during the original eugenics movement.
Next are designer babies, who are babies that are genetically modified using gene editing to have desired intellectual or cosmetic traits. Due to the power of gene editing technologies, we all have the power to create a perfect child in our eyes. And although everyone has a different definition of a perfect child, we can easily have a future where the majority of children are genetically modified to be perfect. The next key effect on the slippery slope is overpopulation. As we start to eliminate disease by using gene editing technologies, more people will live. And while it's hard to argue against people having the right to life, we do need to consider the negative effects that this could have on our world. There could possibly be increased competition for resources, and it might increase climate change. However, all medical innovations throughout time one could argue that those also led to overpopulation. So how much stake should we put in this issue on the slippery slope? Last are irreversible effects to the human gene pool. All our genes are linked in the gene pool, and when one person's genes are genetically modified, it will affect all of us through the trickle-down effect, whether we know it or not. And like I said, these effects are irreversible and unnatural. Once we edit one gene in one human's embryo, there is no going back. We cannot unedit genes. That would just be editing them twice. To wrap up our section about the slippery slope and transition into our case studies, I would like to share a quote from He Jiankui, a Chinese scientist that we will be researching more about in the case study for gene editing. The quote is, "Gene editing for human germline is going to happen in the near future. So I want to remind everyone that we should do this slow and with caution, because a single case of failure may kill an entire field." I find it very interesting the man who ignored all ethical and legal regulations on gene editing and used gene editing to create 2 HIV-resistant humans is even warning us that gene editing is a slippery slope.
So now we're going to transition into our case studies where we hope to hear some thoughts from you guys. Sorry, my phone is ringing. Okay. So firstly, for my case, for prenatal screening, this case is about prenatal screening rates of usage in Iceland. So data released in Iceland has shown that within the last 10 years since prenatal screening was introduced, nearly 100% of women in Iceland who choose to undergo prenatal screening, which is around 85% of the total population, had an abortion after discovering that their embryo had Down syndrome. And this has dramatically affected the Down syndrome population in Iceland. And according to a report from the Icelandic government in 2018, on average, during the past 10 years, only 2 to 3 children have been born each year with Down syndrome in Iceland. So when thinking about these numbers in Iceland, my question for all of you is, do you think prenatal screening should be readily available and accessible to society? And think about what you think the effects of that would be and why or why not you would see it as ethically permissible. So we're going to use the chat feature for this. So if you guys want to take some time to—
Actually, Sami, the chat feature is not working. So we're just going to have to have our guests unmute themselves and respond.
Okay, so if anyone would like to share their thoughts on these questions or this case, just unmute yourselves and share out with everyone.
Hi, this is Jean Kerwin. And I'm wondering if there's any data out there of inquiries made of Down syndrome adults and what their thoughts would be on whether they would have preferred to be genetically screened out or whether they have another opinion about the value of their contributions to society as well as their lives.
I'm not specifically sure in Iceland, but one of my stakeholders in my larger paper is the Down syndrome community itself. And I know that there do exist many organizations of individuals, adult individuals with Down syndrome, who protest the utilization of prenatal screening because they believe that, you know, they contribute to society. So I know in the UK, there were large protests outside of Parliament when prenatal screening became a part of the National Health Service. There are companies such as Don't Screen Us Out, is one I know who I've contacted actually for my research. So I think there do exist a few communities in different countries who are protesting, adults with Down syndrome are protesting the utilization of prenatal screening for this.
Great, thank you.
If anybody else has any thoughts or even questions about this case.
I'm curious to know what the justification the parents had in mind when they decided to have an abortion, when they found out that their kid would have Down syndrome would be, because I'm figuring, but this is just my assumption, that it's more because of that bias, but could it also be because they don't have the resources to take care of a kid with Down syndrome? So I don't know, that's one thing to consider, but it is difficult to come up with like an overall answer to that question.
Yeah, I think it's very interesting because when I studied parents as stakeholders, a big part of that was issues of access and capability, because parents do have autonomy when it comes to their children, and they may not feel like they have the capability or the accessibility to give a child with Down syndrome their needs. But I think also a big part of that is something I studied was misinformation. There seems to be a lot of bias against those with Down syndrome and also misinformation. Through my research, I've discovered how much medical care for Down syndrome has improved, how much more accessible it is. So I think in a way it's a combination of the two. And I think if prenatal screening for Down syndrome continues, in my opinion, there needs to be more education to parents about the modern realities of raising a child with Down syndrome.
Thank you.
Sami, what research has been done, or what do you know about parents who have decided, what conversations have those had, who have decided not to abort a child with Down syndrome?
I know that we actually had some genetic counselors come in and talk to our class, and it was really interesting to hear from them. And I think that slowly genetic counselors' advice is becoming less biased and more supportive of parents, and I think that's amazing, and it's how it should be because it is the parent's choice and whatever they choose to pursue. And I think that's a really interesting question that I haven't done a lot of research on that side of it, and maybe I should. That's a good idea to kind of get both sides of parents' view. But I think it's important to understand that many parents probably are happy that they made the decision they did to go ahead with a pregnancy of an embryo with Down syndrome.
Thanks.
I'm wondering if you learned in your research at all about some of the wider impacts that this might have on the gene pool, because I know that you were talking a little earlier that this could have gene editing and prenatal screening and resulting imbalances in the population could have trickle effects. So I was wondering if you knew anything about how like the elimination of certain, because it looks like Iceland is on that road towards elimination, how that could have an impact on the gene pool in future generations.
Yeah, that's definitely an interesting question. And so far there hasn't been any long-term studies or inquiries into that, of course, because it is quite new. So there is no utilization to have a long-term study yet, but a big part of it that many scientists are questioning are what will the effects be to the lack of diversity in the gene pool. And I think that's really important, but also I can probably speak more to the social side of it. A big part of my research was thinking about what happens when we start to eliminate diversity in our society. How does that change people's views and perspectives on the other? And I think that's really interesting to think about how people are happy to, in a way, eliminate a whole part of our diversity in our population, and how can that lead to a slippery slope of more parts of our diversity in our society being eliminated. Okay, so if anybody else has anything else they'd like to share, we can move on to our second case study about gene editing.
So our second case study is about the experiments of He Zhonghui. In 2018, Zhonghui, a Chinese scientist, and many others were part of a project that used CRISPR-Cas9 to edit out the CCR5 gene. This created two HIV-resistant twins named Lulu and Nana. As you can see in the image, Jean Quay presented at the Second International Summit on Human Genome Editing in November 2018 in Hong Kong, China. At the time, some other scientists praised Jean Quay for his work with gene editing. However, Jean Quay was later tried and convicted of illegal medical practices. More specifically, Jean Quay was found guilty of falsifying ethical review, failing to inform experimental participants of possible risks, and violating ethical guidance. So for Jean Quay's experiments in creating 2 HIV-resistant humans, Jean Quay was sentenced to 3 years in jail, and he is currently serving that time. So there are 2 case study questions I'd like you all to think about, and when you have an answer, you can just unmute yourself and say it. So the first is, what ethical ideas are involved in this case? And the second is, do you think that gene editing should be allowed to be unregulated? And if you think that gene editing needs to be regulated, how do you think it should be regulated? So if you have any thoughts, just unmute yourself now and share them out. And any questions as well. Well, to get us started, I could share what I think gene editing should be regulated. I think that gene editing should be regulated, and it should only be allowed for medical purposes. I feel like there needs to be universal regulations so that there are not different regulations between each country. So ideally, I'd like to have them done by the UN, and there'd be a special committee that would review every case of gene editing, and parents would have to submit a proposal for gene editing for a medical purpose, such to eliminate Down syndrome or HIV. And that would have to be reviewed and approved by the UN. So if anyone has any thoughts on anything involving gene editing or anything in this presentation, you can share them now.
I have a question.
Yeah.
Hello, this is B. Thayer, and I think the equality throughout the world is an interesting idea that you just mentioned, but how about the equality in the cost so that every individual is on the same page if they get to that, whether they're rich or poor, and it comes down to a health system that is equal around the world for individuals?
Yeah, I think that's part of the larger question too of should we have systemized healthcare? And it is a big issue with gene editing because it does cost about $4,500, but it costs more to create the embryo using IVF. So I do foresee as gene editing becomes more available, and if we do decide to put in universal regulations, that the price would be regulated at a much lower rate. And this would allow more people to have access. So regardless of what country you're in and for what reason you would want to use gene editing, I would ideally want the price to stay the same, maybe around $2,000. Although that is still fairly expensive, I foresee that price will continue to drop as more people use gene editing.
Mm-hmm.
I was also just wondering if you've had any discussion about the more social implications of this, because it sort of sounds reminiscent of like what Hitler was trying to do in some capacities of like trying to reform the gene pool. So did you guys talk at all about any of those potential long-term implications?
Yeah, definitely. As Sami talked about with the eugenics movement with prenatal screening, it is a true possibility with gene editing. Because we can create the perfect human, we could have this universal definition of perfect, and we could very easily see future generations that all look the same, all have the same traits. And that would create a new world with a new definition of human. The new normal in a parent's eyes would be a genetically modified human. So this could very easily lead to a similar eugenics movement that we saw in the 20th century. And that could be a very dangerous world that we're leading into.
Thanks.
Do you think that there's a difference in the research around something that has been acquired versus something that's been inborn? When you talk about Down syndrome, that is something that you get, that's something that gets passed on through your chromosomes versus HIV, which is acquired. Do you feel like there's a difference in what you've studied in terms of acceptance based on the origin?
Yes, and part of my paper actually talked about how gene editing for HIV resistance in future generations is a very passive solution to the current AIDS epidemic. It's not really trying to fix the problems of how many people in our world currently have HIV and trying to improve their quality of life. It's more so just preventing it in the future, which isn't really helping anyone who's HIV positive right now. And if Sami wants to speak about Down syndrome, she could.
No, I was just going to add what you said. I think it's really interesting. That's where our topics appear quite similar, but I think that's a point where they kind of go on their own roads, because I think there is a difference. And I think with HIV, where it's acquired, I think what Elizabeth talked about before with the stigma around HIV is a big part of it, because I don't think the same stigma exists in Down syndrome because it is passed down through the gene pool. So people don't really think you got it in the same way. So I think that's really interesting how that impacts society's view on these editing or preventing technologies.
Yeah, and adding on to something that Sami said, many people who are HIV positive, others see them as, oh, they did something wrong in order to get HIV. But if it's Down syndrome, it's just the way that you were born.
Presenters, I just wanted to let you know that you have about 4 minutes left in the session.
Can I ask a question?
Yeah.
So if like one day there's gene editing and that gets to like maybe getting rid of vision impairment or like loss of hearing, could like the potential consequences, aren't that like maybe eye doctors and hearing doctors lose their jobs and don't have like any career and they have to like choose another career?
That is a real issue, and that's something that would be on our slippery slope that we talked about. Just effects to the economy, effects to society, and people losing their jobs because all imperfections were eliminated out before birth. So that's an interesting point.
I just wanted to add something to the economic impacts. You know, we have a lot of safety net programs, huge number through the federal and state governments for people with disabilities, Down syndrome, and other support programs. If we do what Iceland did, or through gene editing, we reduce the number of disabilities, do you think the safety net will disappear for those few who remain unedited?
Yes, I think it would. And as we continue to edit more people and as we continue to use gene editing more and more, we are really redefining what it means to be human. And that'll mean changing all our old ways in the past.
Mm-hmm. I think that's really interesting, because I think with any other presentation you'll see today, that's a big part of all our projects, just our theme of being human in a brave new world. Our world isn't set up for that brave new world yet. So I think with anything that any of the topics we're talking about today, that's a big issue, is all the people who will lose their jobs or current systems that are set up that will potentially fall or not be a part of our society anymore. So I think that's really interesting how it applies to most of our topics today.
Okay, we've got about 2 minutes left, so we'll need to make this one the final question.
Sorry, I guess I could just hear your thoughts with regards to gene editing, about the emphasis that should be placed on quantity or quality of life that is a result of the specific disease that is being targeted. So, for example, with Tay-Sachs, children often don't live until their 6th birthday. So would that be, in your mind, more or less ethical than other diseases or HIV elimination?
Yeah, that's something that I looked at very early on in my project, how gene editing for some could provide true life because you'd be eliminating out a fatal disease like Tay-Sachs. And then gene editing for HIV resistance is more so just to prevent the possibility, However, there is still the possibility that someone who you spend a child who is genetically modified to be HIV resistant would never even come in contact with HIV positive blood. So it's really not bringing them any benefit to be genetically modified. So I feel like as gene editing becomes more prominent, we would probably see a spike in gene editing to eliminate fatal diseases more so than resistances like HIV. So I think we are going to transition into our final slide and just say thank you so much for coming to our presentation. If you have any more questions, you could always email us. I'm Elizabeth Weichner.
And I'm Sami Sharp.
And we're so happy that you all came. So thank you so much.
Thank you. We hope you enjoyed.
Feel free to unmute for some applause if you'd like.
Yay, good job.
Great job.
Wonderful work, girls. And we will see you in the next session. To our guests.
Thank you.
Yeah.